ample evidence that there could be autonomic nervous system disorder going on as well. What I can tell you, and it remains to be determined whether it's going to manifest itself within the heart. The mouse demonstrates an autonomic nervous system issue that manifests in dilated cardiomyopathy. Humans remains to be determined. Yet there is strong evidence for autonomic enteric nervous system issues in type 1 SMA kids. What we know is a systemic gene delivery targets not all autonomic networks, but many, at least in the mouse, the ones that we've studied more intensively that are connecting to the heart. So we can correct most of the dilated cardiomyopathy by a systemic gene delivery. We do not appear to correct it at the same level in the mouse by an intrathecal delivery. That doesn't say that we're not generating positive preclinical data by intrathecal delivery. It has just not been as robust as the systemic gene delivery. We do have plans to move forward with an intrathecal gene delivery program as well, so there is likely going to be another RAC submission by this team if things continue to move forward. So I think we're paying attention to this, and really we will be positioned, if successful, to really be evaluating in a near head-to-head comparison of the same vector production style, purification, same vector itself, with two delivery paradigms, ultimately in hopes to choose and result in the best clinical meaningful outcome for patients. Again, first off though, we can also be addressing safety issues, which is first and primary.
How about redosing?
For vector redosing, and we can certainly, it concerns me on the aspect of the immune system and development of neutralizing antibodies. So there is likely a window of opportunity for redosing that remains to be determined in any of our preclinical studies with an AAV9 vector. Certainly one could then think about immune suppression, but I think that also concerns me. These are, in a type 1 patient, these are weak children, and I certainly get concerned about the thoughts of putting a type 1, I'll let the clinicians speak to this if they want, but I suspect they agree with me. SMA babies we likely do not want to treat them with immune suppressants. Certainly not for long.
I can tell you that for the last 3 years we've studied redosing extensively in the non-human primate with AAV8 antibody. And we have done repeated plasmapheresis in those monkeys and have had incredible success. We actually had better success with plasmapheresis in a window of time for achieving a high transduction efficiency than we did with immunosuppression compared to prednisone and triple therapy. And we're just about to publish that data now. So I think that'll be a very important contribution.
Any other comments? I think I have some.
Yuman?
And I just want to say that it's heartwarming to see how hard you and your team have been working and are continuing to work for this group of patients and their parents that clearly deserve it and more. So I'm very, very pleased to see this study go forward. I just wanted I realize that you will be, once you have this dose escalation study, the higher dose, you will be at the limit of the amount of DNA that you can deliver. But since I continuously think about DNA making RNA making protein, the end product is the proteins. I was wondering whether, first of all, whether you could be studying what kind of protein expression you get in the individual cells, and also whether you could possibly do better at the level of RNA stability, RNA translation, because there are things that can be put into this gene block for the future that could, push that in future trials to a higher level, even if you can't increase the number, the vector dose.
Arthur, you want to take a stab at that one?
Yeah, I'll take a stab at it. I think, first of all, it's if you move SMN levels up even a little bit in the motor neuron in the spinal cord, not very much, you get a major impact. So, for instance, one of the mice lines that we made that I say has leaky expression had approximately the level was 3 times over the SMN2 basal level. So you've got 2 copies of SMN2, 3 times the amount, and that was very difficult to measure. So it could be 2.5 times versus 3 times. So actually not very much. Had a major impact on the mouse going to 150 days. So I think it's not so much how much SMN you produce, how much SMN you're producing from a vector. It's how many vectors you get in each cell that is really the critical issue. So if you can enhance the biodistribution of the vector, that would make it even better. The intrathecal delivery that we've done in pigs, you can get 80% transduction in the lumbar cord. It's slightly less as you go up to the cervical cord, even though you've gone into the cisterna magna. So it doesn't fully resolve the issue of getting maximum distribution of the AAV.
You need to get more cells that actually
Yes.
get one copy of the
Yes.
vector, not more
Exactly.
copies won't really
Yeah.
help you.
Yeah.
Okay.
Dr. Foss?
Yes, just another question about the alternative sections on the consent form. You don't mention home ventilator programs, which there are several centers in the U.S., and I'm wondering if you, first of all, have such a program at your institution, or B, if there's some reason you didn't mention to the potential subjects of that as an alternative.
Well, that's an ongoing, I'll let John answer that, but that is an ongoing program that we...
Yes, we do have a home ventilator program. Richard Schell, the pulmonologist, works in our SMA clinic. He's excellent at that. But I wasn't sure I understood the context of the question. You mean as an alternative to being in the study?
In the alternative section of the consent form, there's no mention of it, so I don't know how families would necessarily, I mean, maybe they know anyway, but it seems to me it ought to be listed in the alternatives.
I see. Yeah, actually, that's an excellent suggestion. We can put that in there. We do discuss that with all the patients at the time that we see them, if it's appropriate, if the parents are ready for that. Yeah, an excellent point. And so the standard of care document that Chin Wang was first author on, we sort of scrupulously adhere to that. I was discussing that with some of the parents today, and that'll be a major part of this protocol. And the standard of care, of course, will involve respiratory support, either through BiPAP or invasive ventilation, if needed.
Any other RAC member comments? Yep. Dr. Cohn?
So I have a question, a couple of comments. The question is, what do you know about transplacental transfer of antibody to AAV? You must have looked in your previous infants. I assume most mothers have high levels of AAV. How much is transferred to infants? So how many will or will not be eligible based on experience?
So first, AAV, I think to back up and to say that AAV2, which is the most predominant serotype, the numbers are quite high for the number of individuals who have high neutralizing antibodies for adults who have high neutralizing antibodies against AAV2. For AAV9, Jim Wilson's group has published within the United States, there are approximately 15% of adults that would have neutralizing antibodies that would be of concern. We certainly, our group has talked about, so is there a chance of placental transfer of neutralizing antibodies? And the answer is absolutely yes. We are also going to be checking the babies for that, and I think we've discussed about even checking for the parents, for the mother as well. Certainly we will be checking the child.
Right. No, I was just concerned that it would be higher and that you would rule out a lot of patients. So if it's 15% roughly, and it's a fraction of those, that's good.
Yeah, just one quick comment. We do screen all our primates for anti-AAV8, anti-AAV9 antibodies, and what we see uniformly is actually in the newborns, there's almost no seropositivity in those newborn animals. And so actually it's kind of a paradigm that transplacental transmission actually is not thought to occur very efficiently for whatever reason, because of all these newborn primates that when they're screened you just don't see binding antibodies to AAV.
Okay, and then a comment was Dr. Choka's concern about possibly early termination of the study because of SAEs. And would it be possible to talk about only product-related SAEs as being ones that would call stopping rules? Obviously, you need to record all the AEs, but if they're only the ones that you are probably product-related might be considered, I suspect you won't have a lot of those as opposed to the more common ones related to the disease.
That's a good point. We'll include that.
And then my final comment is that I would also agree that I would like to see us put in our recommendation to consider starting at what you think is a therapeutic dose rather than doing a classic dose escalation. It's different when you're using a new chemotherapy agent in adults with advanced cancers as opposed to children with rare inborn diseases where they may have only one opportunity to get treated. And so,
the only comment is I wish you had been at our pre-IND meeting.
I've been there, but on the other side.
Okay. Any other comments from the RAC? Comments from the public?